Hello, everyone. Welcome. Happy Thursday. We're just a minute or so before 3, so we'll let people join in. Go ahead and type your first name or your nickname into the chat for me, so I have a record that you were here today. Thank you. Thank you very much. Just about everybody's here, but we'll give people another minute. Very good. I hope you're having a good day so far. Hope the week is going well for you as things get started up again. Yes, just go ahead, if you would, and type your name into the chat for me, or your nickname into chat for me. That'd be great. All right, very good. Okay, so the last time we were together on Tuesday, we talked about the course and how the course is organized, and we started our first lecture topic, which is the universal characteristics of cells. And what we're going to be doing today is finishing up that lecture, essentially. You'll notice on our schedule for this week that there's a second lecture about the organelles. What I'm going to ask you to do is to watch that lecture on video from our playlist on YouTube. That lecture actually has three short videos in it, and it's quite straightforward. It's about the organelles that we find in eukaryotic cells. It's a description of each organelle in terms of what it looks like, how it's structured, and what it does, what its function is. So it's a very straightforward lecture, and I think you can handle it from home quite well. Be sure to take notes for yourself, however you want to do that. And that will finish us up for the week. You do have a lecture assignment this week, which is to outline the first 2 lectures. I'm going to go ahead and pull up, I'm going to share my screen with you. And I'm going to pull up Canvas for us for just a minute. That's what you should be looking at right now. Oops, hold on. There. That's what you should be looking at right now. You should be seeing the Canvas course, BIO312, and I've just clicked on modules over here. So I'm going to scroll down to this week. Here's our to-do list for the week. We started into this lecture when we were together on Tuesday. Here's the one that you'll be watching from home. So here are the slides. These are the slides for this lecture. And then what you'll need to do is just go to our playlist on YouTube in order to find the video. So let me pull that up for us for just a sec. Pull this up here.
Is the outline on both of those links for the slides, like the universal characteristics of, and like the biochemistry?
Organelles, the organelles one. So the
Okay.
first lecture is universal characteristics of cells and biochemistry. We'll finish that today together because that one's a little more complex. So I want to make sure we go through it together. And then the second lecture is called Cellular Organization and Organelles. And essentially what it is, like I said, a description of each of the organelles, what they look like and what they do.
Okay, I was just confused because we didn't get through the first one yet. So I didn't know.
Okay. That's right. So we'll finish that first one today together. So what you should be seeing right now, Let me just make sure it's shared. What you should be seeing on your screen right now is our playlist up on YouTube. You can see cell biology, BIO312, and it's loaded up the lecture we're doing today. But over here, this is the list of all the videos that's in that playlist. And you can see there's one called Organelles. And it has 3 separate videos in it to make the one lecture. Okay, so this is the one that you'll want to watch from home. And again, take notes for yourself. And then for the assignment, let me pull this assignment up so we can see what it says together. Whoops, let me get rid of this one. I keep trying, there we go. So you should be seeing again our modules page. If I click where it says lecture assignment week 1, if I click on that link, these are the instructions right here. And you can see they're not very extensive. It just says your assignment for this week is to outline both of these lectures. You can create a rough outline, remember, directly on PowerPoint. And then copy and paste the outline into a Word document, and then customize it, change it according to your own needs. How do you want your outline to look? I'm not looking for you to submit outlines for these 2 lectures that you think I'm going to want to see. I want you to make 2 outlines that you find useful for studying. Okay, now we said you're also perfectly able, if you want to, you're perfectly welcome to write outlines by hand, if that is more helpful to you, if you would rather have a handwritten outline. It's all up to you. You are making these outlines because they're going to help you when it comes time to prepare for the exam. It's going to give you a way to study these 2 lectures in outline form. If you are submitting handwritten outlines, what you're going to want to do is take pictures of those outlines, try to get them as clear as possible, please. And then insert them into a Word document. Okay. Remember, you have access to Microsoft Office from the Keene State website, and you can open up Word from there. And you can create whatever documents you want in there. And then you'll just submit your document through Canvas. And I'm just looking to have those in by Sunday at midnight. Okay. So when you're making the outlines, make them according to your tastes, according to what you think is going to help you learn the material. The reason I didn't give you very specific instructions about how to make them is because everybody, learns differently. Everybody finds a need to put different things into their outline. I just want to encourage you to make outlines because I think it's a good way to manage all of the material that you're going to need to learn for the exam. Okay. Does anybody have any questions about that? Okay, good. All right. So what we're going to do now is pull up the slides from the lecture we started on Tuesday, and we'll jump in where we left off, which is right around slide number 11, I want to say. Okay. So do me a favor, if you would, go ahead and turn your camera on and leave yourself muted. You're welcome to unmute if you want to ask a question, or you can type your question into chat. Either way, that's fine. Okay. And we'll go from here. So if you remember, the last time we were together, we started talking about these, universal characteristics of cells. And we finished up last time on slide number 10. We were talking about some of the work that's been done to try to explain how cells might have first emerged, how life might have first emerged on planet Earth. And we talked about this finding that the first life form may have actually been a ribozyme. And remember, we said a ribozyme is not a cell, but rather a piece of RNA that's capable of copying itself. We know there are still ribozymes present. We know that there are ribozymes that are functional in living cells. So the idea is that perhaps a ribozyme emerged or evolved, and then later on it became surrounded by a membrane. And that would have made it a very primitive form of a cell. Either way, we now estimate that the first cell probably appeared about 4 billion years ago. The most recent research suggests that the very first, very primitive cells probably emerged about 4 billion years ago. Remember, the planet was covered in water at that time. So the first cells would have been in the ocean. Now, what I want to talk about next is a little bit about cells and energy. We're going to spend a whole day talking about energy later in the semester, but for today, I just want to talk about cells and their need for energy. Remember, we define energy. We define energy as the ability to do work. So when we're talking about cells needing energy or using energy, what we're really talking about is cells doing work. And for a cell, work is primarily building things, breaking things down, or moving. That's essentially what's happening in a cell if they're capable of movement. And in order to do work, there has to be an energy source available to them. I got a message from, is it Kylie? I got your message, Kylie, so no problem. All right. Now, we mentioned last time that there's a molecule called adenosine triphosphate, or ATP, which is sort of this universal, we refer to it as currency, cellular currency. It's a molecule that cells use when they need a source of energy. And that's because ATP has covalent bonds in it. It's a group of atoms that are all bound together covalently to make this molecule that we call ATP. And when you break specific bonds in that molecule, a bundle of energy is released, and the cell can use that energy in order to do work. Now, ATP comes from somewhere. It's built, it's made during a certain metabolic reaction, and it depends upon the type of organism that you're looking at as to what kind of a reaction is making ATP. For example, in humans, our cells are making ATP primarily by using what's called cellular respiration and oxidative phosphorylation. That's actually a long, long series of reactions that breaks down glucose down into carbon dioxide and harnesses all of the electrons that were released from the glucose molecule and uses the energy from those electrons to build ATP. Our cells are also capable of a metabolic reaction called fermentation. We can use fermentation as a secondary way to make ATP when our cells are not able to do cellular respiration because there's not enough oxygen around. We do a type of fermentation, remember, called lactic acid fermentation. other organisms that can ferment do different types of fermentation. The thing to remember is that cellular respiration and in all of its forms, there are two different types of, or I should say multiple types of cellular respiration. This is a very efficient, a very good way to make ATP. Fermentation is a lousy way to make ATP. It's just not very efficient, but it still is a way to make ATP. So if a cell can't manage cellular respiration, it will gladly do fermentation because cells cannot survive without ATP. ATP is a source of energy for the cell. We think of it as energy currency. It's actually used for what we refer to as energetic coupling. Now, again, we're going to talk about this in much more detail later in the semester. But for now, understand that what cells do with ATP is they break it and then they build it, and they break it and build it and break it and build it. When it's produced through cellular respiration in large quantity, a cell can break one of the phosphate groups off of ATP and release energy that it can use to do work. When there's energy being released in other places in the cell can harvest that energy. It can store it by putting that phosphate group back on to ADP to make ATP. So one of the concepts we're going to talk about later in the semester is this idea that there's a difference between how a cell makes ATP, that's cellular respiration, and how a cell uses ATP. Cells make ATP using that metabolic reaction, that series of reactions, cellular respiration. But they use
Professor?
Yes.
So just kind of clarification, when you said that like the ATP is the energy we get is when it's broken, so it's sort of like nuclear fission when they break the uranium, and that creates heat and energy, and that's how we generate electricity. So it sounds like that's pretty much very similar to how ATP works.
That's a good analogy, in that when you break, of course, in fission, we're talking about breaking an atom, but in with ATP, we're talking about breaking one of the bonds in a molecule. What a covalent bond is, the bond that holds atoms together in a molecule. What a covalent bond is actually a pair of electrons and their energy, the energy that they have within them as part of them. So whenever a covalent bond gets broken on a molecule, energy is released. Cells are funny, cells are thrifty. You'll hear me say that again and again. They don't waste. So if a molecule is being broken down and bonds are being broken, energy is being released out of those bonds. And the cell does not want to lose that energy, even if it doesn't need it right now. Even if it has no need for it right now, it's going to want to grab it. It's going to want to harness it. And it does that by storing it in ATP. Then later on, if it needs energy, it can break a bond in ATP and get it back out. So little ATP gets broken and then rebuilt, and then broken and then rebuilt, and then broken and rebuilt all day long. That's what we call, coupling, energetic coupling.
Didn't you say yesterday or the day before that cells only make ATP when they need it? They don't like make, so how do they store energy by making ATP if they only make enough when they need it in the moment?
Yeah, this is where this language comes in. I want you to make note of. And again, we will talk about this at a lot more detail later, but there's a difference between how a cell makes it, produce, and how a cell uses it. Part of the use of ATP is to take the phosphate group off. When it takes the phosphate group off, the outer phosphate group, because remember, it's a triphosphate. It has 3 phosphate groups on it. When it takes off one of those phosphate groups, a little bit of energy is released. When a cell has a little extra energy around that it doesn't want to lose, it puts the phosphate group back on and stores that little bit of energy in that bond. So making ATP is what happens in cellular respiration. ATP is made from scratch, if you will, during cellular respiration. Cells will only do cellular respiration when they need to make ATP. But while ATP is made and in the cell, they're going to use it by taking off the outer phosphate group and putting it back on, and taking it off and putting it back on, and taking it off, releasing energy, putting it back on, storing energy, taking it off, releasing energy, putting it back on, storing energy. It's a very strange concept, I know. But that's all you need to know for right now, is that the way a cell uses ATP, this taking off, putting on, we call coupling. We call energetic coupling. Okay. All right. Now, the way that a cell, I said that whenever a cell breaks a bond, whenever enzymes within a cell break down a molecule during a chemical reaction, they're actually breaking the covalent bonds that hold that molecule together. And there are certain types of organisms that harvest energy like this, that harvest that energy that's being released from that broken bond using different materials. I have 3 different categories here, 3 terms that represent 3 different types of organisms based upon the molecules that they break down in order to make ATP, in order to do cellular respiration, in order to do fermentation. You and I, for example, and other animals, we use glucose. We use a carbohydrate called glucose for cellular respiration. And the long series of reactions that occur during cellular respiration involve breaking glucose apart bit by bit by bit. There are lots of bonds in glucose, lots of covalent bonds. And as each of those covalent bonds is broken, the electrons that sit in those bonds get released. So the question is, do all organisms use glucose? And the answer is no. There are different types of things that organisms, I'll say, eat in order to do cellular respiration or fermentation. There are organisms that use glucose like us, and there are organisms that do other things, and we can categorize them based on that. So the first term here is organotroph. The other term you'll hear is heterotroph. Those mean essentially the same thing. Then there are phototrophs, and the other term that you'll hear is autotroph. Those mean the same thing. And finally, there's a group called the lithotrophs. So let's compare them. An organotroph, or a heterotroph, is an organism that gets glucose by consuming other organic compounds. Then they're going to take that glucose molecule and they're going to break it down or oxidize it in order to make ATP. That's cellular respiration, essentially. So if we call something an organotroph, if we say that organism is an organotroph, the word organic in that word really means carbon, carbon containing. That's the scientific definition of organic, carbon containing. So if we call something an organotroph, what we're saying is they eat carbon, and they eat it, again, for us in the form of glucose, by consuming other organisms. An organotroph is going to eat other organisms in order to get the molecule, glucose, that they're then going to take apart in cellular respiration in order to build ATP. So you and I are organotrophs. Other animals are organotrophs for the most part. Now, the second category is called a phototroph or autotroph. Phototrophs actually don't have to eat in order to get their glucose. They don't have to. They're going to make their own glucose by harvesting energy from light. So what we're talking about with a phototroph or an autotroph is an organism that can do photosynthesis. Photosynthetic organisms are able to harvest light energy and use it to build carbohydrate, primarily glucose. Then they turn around and they do what we do, which is break down that glucose so they can make ATP. So organotrophs, we have to eat our glucose. Phototrophs don't. They can make their own glucose. And then we all oxidize or break apart glucose to make our ATP. Now, the last type of organism is a lithotroph. And what a lithotroph can do, they also have to eat like we do, but instead of eating things that contain glucose, they eat inorganic material. Lithotrophs are fascinating. There aren't that many of them, or at least there aren't that many that we know of yet. But lithotrophs can consume inorganic molecules. Inorganic. So I'm talking about things like, rock and sand, things that don't contain carbon in them. They can eat those things. And instead of using glucose, instead of breaking apart glucose to make ATP, they break apart those inorganic molecules to make ATP. Very bizarre, much, much different than the organotrophs. Most of the organisms that we talk about, including things like bacteria, are organotrophs. They're using carbon-containing food in order to get some glucose that they can then burn or take apart in order to make ATP. We're going to talk about all the gory details of cellular respiration and fermentation a little bit later in the semester. The other characteristic of living cells is that they use DNA as their genetic material. So it doesn't matter what kind of cell you're talking about. It doesn't matter if it's a bacterial cell or an archaeal cell or a eukarya cell. All of them use DNA as their genetic material. And remember, DNA is simply code. DNA is a molecule that stores information. And the cell has to then decode the DNA in order to build protein. And that leads us to the statements of what we call the central dogma. The central dogma, which is sort of our base foundational understanding of how information flows from the DNA molecule into all of the different traits that a cell has, all of the different structures and functions that keep a cell alive. So there are 3 statements to the central dogma, remember. DNA is transcribed to RNA to make RNA. That process is called transcription. RNA is translated to make protein. That process is called translation. And it's protein that gives us our traits. It's protein that gives cells their structural and their functional traits. So the central dogma essentially tells us how genes are expressed. Because remember, it's genes, individual genes that code for individual proteins. Genes are pieces of DNA, lengths of DNA that contain the code to build a single protein. So when we talk about the central dogma, transcription and translation, we're really talking about how genes are expressed, how DNA is used to make protein. It's important to remember, though, that the DNA molecule is used for another process in the cell that's equally important. And that's called DNA replication or DNA synthesis. Whenever a cell prepares to divide, whenever a cell gets ready to divide, it has to copy its entire DNA molecule. And that's because it needs to give each of the new cells, each of the daughter cells, their own copy. So it takes its entire DNA molecule and it replicates it, it copies it. And then each of the new cells will get their own copy. So DNA is actually used for two different things in a living cell. It's used to make more DNA, and it's used to make this molecule called RNA during transcription, during gene expression. This little diagram down at the bottom of the slide just shows us that in a simple form. First, we can see that DNA is used to make more DNA. That's what that purple arrow is showing us. That process is called replication or synthesis. And the other thing that DNA is used for is gene expression through transcription and translation. DNA is used to make RNA through transcription. And then RNA is used to make protein through translation. One of the things that is sometimes confusing to people who don't study science is the idea that all cells, we said all cells use DNA as their genetic material, and all cells contain the entire DNA molecule for that organism. That's a strange concept for people. If you talk to people who are not scientists, there's an understanding that somehow the DNA in, let's say, a skin cell is different from the DNA in, let's say, a liver cell. People think, well, those are two different cells, so they must have different DNA in them. In reality, the entire DNA molecule is present in both of those cells. Whatever that organism's DNA molecule looks like, it's present in both of those cells. The difference is that only some of the DNA molecule gets used, gets expressed in a skin cell versus a liver cell. And that's why those two cells look and act so differently. The skin cell is going to use the genes that produce proteins that a skin cell needs. The liver cell is going to express the genes that make proteins that a liver cell needs. So each cell has its own set of genes that it's going to express, and it's going to leave the rest of the genes alone. It's not going to turn those on. It's not going to use them or express them. There are lots and lots of genes in the DNA molecule that never get used. That's another strange concept. The idea that there's lots of DNA inside of human cells that will never be used. It will never be expressed. You have to remember that the cell theory tells us that all cells come from previously existing cells. That means that the DNA molecule that we have today in all of our cells, we actually inherited from our very most ancient ancestors, our very most ancient cellular ancestors. And we have remnants of that DNA today. We have genes in our DNA molecule, for example, that code for the proteins to build gills. We don't need gills. We don't extract oxygen from water. We can't anymore. But there was a time in our evolutionary history when our ancestral organisms expressed those genes and created gills for themselves. We have DNA in us to build a tail. We don't express that DNA anymore either, at least not usually. Every once in a while, a baby will be born that has a little tail. And that's because that DNA was accidentally expressed during fetal development. And yeah, every once in a while, some of those genes get turned on by accident. But yeah, our DNA is full of genes that we don't use anymore. It's very strange. It's a strange idea, but it's absolutely true. Now, there are some genes that every cell uses. We often refer to those genes as housekeeping genes. Let's think about what they might code for. Remember, all cells in, let's say, the human body, all cells have certain jobs they have to do every day that require particular enzymes. So the genes that code for those enzymes are used in virtually every cell. Now, there is sort of an ongoing 24-hour-a-day need for those enzymes. So there are some genes that you find being used or expressed around the clock. The need for that protein is so great that the cell makes it around the clock. Any gene that gets transcribed like that, gets expressed like that continually, we refer to that as constitutive expression. Any gene that a cell is going to use just constantly, it's going to express it constantly. It's going to make protein from it constantly because it has an ongoing need for that protein. 24 hours a day, 7 days a week, 365 days a year. We say that that gene is being expressed constitutively. It's constitutive expression. It's always, in other words, turned on. That gene is always on. It's always being used. There are lots of other genes, though, in the DNA that are not used all the time. In fact, they're only used at very particular times. We refer to that kind of gene expression as regulated gene expression. Cells have the ability to turn on or use or express a gene when they need it, and then they can turn it back off. They cannot use it for long periods of time. That's referred to, again, as regulated gene expression. Some genes are always on, constitutive expression, and some genes are regulated. They're only used periodically when the cell has a specific need for that protein. We're going to switch gears now. We're going to leave the topic of cell structure and cell function for the rest of this lecture. And we're going to talk instead about a little bit about biochemistry. We're going to do a little bit of a review of biochemistry. Now, whenever I say that to students, sometimes I see them grimace a little bit We have some chemistry majors in here, which is great. Don't be afraid of chemistry. There's certain chemistry that relates directly to biology, and that's the kind of chemistry that we need to review, that we need to have a good grasp on, because it plays such an important role in our understanding of cell biology. And that's what we'll review today. I have a question from Cecilia. In my cell bio, my 111 class, which was the evolution class, is that what you're talking about? We saw early developmental pictures of, go ahead.
I don't remember what it was. It was at a different different
Oh,
school,
okay.
but it's probably very similar. It was like introduction to cell biology and evolution, and it was kind of a whole bunch of everything.
I think I know I know the picture you're talking about. The picture, it's a series of images of developing embryos or developing fetuses. It compares, you've got fish, chick, human, and bat. It's 4 very different organisms that you would not think were similar to each other. And it's a point during early development when the embryos look very similar. And this really freaks people out. There's a stage in human development, in the human embryo, where the gills are there. There is a stage in human development where humans have gills. Now, those gills don't stay there, obviously. They are then systematically removed from the embryo. But yeah, in early development, in fetal development, there are some genes expressed that absolutely come from ancestral organisms from millions of years ago. If you look at a chick embryo and a human embryo, especially side by side, you'll be hard pressed to tell them apart. Yeah, it freaks people out when you tell them that. What do you mean? What do you mean a human embryo, has this structure on it? Yeah, it's very strange. But you have to remember that we inherited our DNA. We inherited our DNA from all of our early ancestors. It's been passed down for millions of years. And because of that, we still have a lot of DNA that we just don't use. But an related organism did use many, many years ago. All right. So for some of you, the rest of the lecture today is going to be complete review. It's going to be very basic. You'll feel like you don't even need to hear it. But for others of you, this might be an important review because maybe you don't remember this. You don't recall this information. The idea is that it'll help all of us get on an even footing for the kinds of chemistry that relate directly to biology, the kinds of ideas and terminology that we need to have a good understanding of so that we can talk through cell biology for the rest of the semester. So the first thing I want to do is just review a couple of terms. Remember, there's a difference between an element and an atom and a molecule. Remember, the elements are those things that can't be broken down anymore, and they can't be converted into something else. So we go to the periodic table of the elements, and we can see all of the known elements in that table. And each element is slightly different. It has slightly different atomic structure. An atom is just the smallest piece or particle of that element that we're going to find in a cell. Now, yes, there are subatomic structures. And if you take chemistry or physics, you'll learn about those. But the atom is the smallest sort of piece of any element that we're going to encounter in a living cell. When atoms are bound together, you create a molecule. So it's important that we keep atom and molecule separate in our heads. An atom is that single unit of a particular element, and a molecule is a combination of atoms. We build molecules with covalent bonds. And the last term I want to just review is this word polymer, because I tend to use it when we're talking about macromolecules and things. There are molecules in cells that they're polymers. They are repeating units of monomers. So, for example, I'll often refer to polypeptides or folded up proteins as polymers because even though they're not true polymers, they really aren't. What we have inside of a protein is just a long, long string of repeating subunits. And those subunits are amino acids. When you join amino acids together into a long chain and then fold it up, you're going to get a protein. So you'll hear me using that word, like that polymer word, to try to drive home the idea that some molecules are built out of long, long chains of repeating subunits. So to review a little bit about atomic structure, remember when you go to the periodic table, every type of element, it has a name, it also has a symbol, it has a lettered symbol. And then there are a couple of important quantities associated with that element. So for example, this is taken off of the periodic table for the element called carbon. Carbon, the symbol that we use for carbon is the capital C. And periodic tables will arrange these numbers a little bit differently. It depends on the table that you look at. But it's going to give you both an atomic number and an atomic weight. So it's going to give you two numbers associated with one atom of this molecule. So when we talk about an atom of carbon, for example, the atomic number is the number of protons that are in that atom, 6 for carbon. The atomic weight, or what is sometimes called the mass number, is the number of protons plus the number of neutrons. Remember, every atom has an atomic nucleus where the protons are and the neutrons are. And then it is surrounded by a cloud of electrons. If it's a neutral atom, if it doesn't have a charge on it, the number of protons and the number of electrons is going to be the same. If it's a charged atom, it's going to have an unequal number of protons and electrons. So for carbon, there are 6 protons in carbon. We know that the number of protons plus the number of neutrons is 12, at least in the most common form of carbon found on our planet. So if there are 6 protons and the number of protons and neutrons is 12, then that means the number of neutrons is also 6. 6 plus 6 is 12. If there are 6 protons, we know there are also 6 electrons in that neutral atom. One of the things we talk about a lot in biology is molecular weight, because it's going to influence, for example, what kinds of molecules can come in and out of a cell, can come across a cell membrane. The molecular weight, the sum of the atomic weights of all of the atoms that are in that molecule. That's all molecular weight is. We are taking an atomic weight of each of the atoms in that molecule, and we're adding them up. So, for example, a molecule like water. Water is H2O. It has 1 oxygen atom in it and 2 hydrogen atoms. So if we want to know the molecular weight of water, we go to the table, the periodic table. We need the atomic weight of oxygen. We need the atomic weight of hydrogen and the atomic weight of another hydrogen. And we're going to add them up. And that's going to be the molecular weight. All right. Sometimes when you hear molecular weight, you kind of go blank and you think, oh, I don't know how to calculate that. You really do. You're figuring out the weight of a molecule by adding up the weight of all the atoms in that molecule. Now, the other thing that we deal with in the laboratory anyway is needing to produce solutions from different molecules. And that's where molarity comes in. A lot of the solutions that we use are a certain molarity. And of course, the technical term or the technical definition for this is the number of moles of a substance per liter of solution. And remember, moles are just molecular weight in grams. So for example, when a substance has a molecular weight, let's say of X, then 1 mole of that substance is going to be X grams. If I want to make a 1 molar solution, I'm going to take those X grams and I'm going to put them in 1 liter of whatever it is I'm dissolving it in. Let's say I want to make a 1 molar solution of glucose. That means I need 1 mole of glucose in 1 liter of solution. So I need to know the molecular weight of glucose, and it happens to be 180. So a mole of glucose is 180 grams. The molecular weight in grams. I'm going to take 180 grams of glucose and I'm going to dissolve it in 1 liter of water. And I'm going to have a 1 molar solution. You should know how to do that. You should know, if I ask you what is a mole of glucose, you should be able to, if you had the periodic table, you should be able to tell me that it's the molecular weight of glucose in grams. And then if I wanted to make a 1 molar solution, I would have to put that in a liter of some kind of solvent. For glucose, of course, that would be water. Now, one of the nice things about biology, I will say, about cell biology, is that there are really only 4 elements that we deal with the most. There are 4 what I call critical elements in living cells: carbon, hydrogen, oxygen, and nitrogen. If you take all of the living cells on our planet and you break them down into all of the different elements that are inside of them, 97% of that material is going to be carbon, hydrogen, oxygen, and nitrogen. So yes, of course, cells have other elements in them. We have sodium in us, we have chloride, we have potassium, we have magnesium, we have all different kinds of things in us. Of course we do. But 97% of living material has 4 elements in it: carbon, hydrogen, oxygen, nitrogen. That's it. That's it. We're actually pretty simple. We're actually pretty basic when you think about it. Now, something related to chemistry that's going to be important for us to know is this concept of electronegativity as it relates to those 4 critical elements. Now, electronegativity is a characteristic that each different element has. All it is a measure of how well that element can attract electrons to it. All right, that's how I want you to think of electronegativity. It's a measure of how well an element can pull electrons towards it. And if we take our 4 critical elements and we put them in a scale of electronegativity, it looks like this. The most electronegative is oxygen. And that's followed by nitrogen. And that's followed by both carbon and hydrogen. Carbon and hydrogen have approximately the same electronegativity. So in biology, at least, we think of them as having the same electronegativity. Oxygen is the highest, then nitrogen, then both carbon and hydrogen. That means oxygen has the ability to pull or attract electrons much more than carbon does or hydrogen does, even more than nitrogen. Nitrogen sits below oxygen. It's less electronegative than oxygen is, but it's more electronegative than carbon and hydrogen. Now, why do we care how well an element can pull electrons towards it? We care about that because those four elements are going to be joined together in molecules inside of cells. They're going to be joined together with covalent bonds to build molecules. Remember, a covalent bond is simply a pair of electrons that are being shared by two atoms. So if one atom has an electronegativity that's much higher than the other atom, what's going to happen is those electrons are going to move in that bond towards the more electronegative atom. In other words, one of those atoms is going to hog those electrons. It's going to pull the electrons very close to it. And the other poor element, the other poor atom, is going to be left very far away from those electrons. And we care about that because that's going to change the way that molecule behaves in the cell. So electronegativity really does matter inside cells because molecules will behave differently depending on the electronegativities of the atoms that are inside that molecule. Inside of an atom, the protons and the neutrons are clustered together in the atomic nucleus. The electrons are sort of flying around the atomic nucleus in a cloud. Remember that we can actually make predictions about where those electrons are going to be at any one moment in time. Even though it might look to us as though the electrons are just buzzing around randomly around the atomic nucleus, the truth is they spend most of their time in certain locations. We refer to those locations as orbitals. And orbitals get organized into a level or a layer called a shell. So around an atomic nucleus, there are shells that contain these orbitals that contain electrons. Now, why do we care about that? Well, the outermost shell, the outermost location of electrons around an atom is called the valence shell. And sometimes in the valence shell, there are going to be unpaired electrons. Sometimes in the outermost shell around an atom, there are going to be unpaired electrons. That's called the valence shell. We care about an unpaired electron because an unpaired electron is unstable. So any atom that has an unpaired electron in its valence shell is an atom that is ready for a chemical reaction. Nature seeks out stability. Nature likes electrons to be in pairs because when electrons are paired, they are stable. An unpaired electron needs a mate. And the way it's going to get a mate is through a chemical reaction. One of the most common ways for an unpaired electron to get a mate is to come together with another unpaired electron. In other words, if one atom that has an unpaired electron in its valence shell comes together with another atom that has an unpaired electron in its valence shell, those two atoms can share those two electrons. Now they both have a pair. They each brought one. They created a pair, and now they're sharing that pair. That is a covalent bond. That's all it is. It's a pair of electrons that are being shared between two atoms. That's all it is. So when we say, oh, there's a covalent bond between an oxygen and a hydrogen, what we're really saying is that the oxygen and the hydrogen are sharing a pair of electrons in their valence shells. And when they share those electrons, it makes both atoms more stable. That's why covalent bonds are so strong. That's why they're hard to break. A cell has to do something to break a covalent bond. It has to invest some of its energy into breaking apart a covalent bond, because the two atoms that are in a covalent bond are much more stable together than they are apart. So the reason molecules hold together so well is because they're made from these covalent bonds. And it's the covalent bond, the pair of electrons, that are holding those two atoms together. In this little diagram down at the bottom, we see a covalent bond forming. Here's an atom. I don't know what it is, but here's an atom. Here's another atom. They both have an atomic nucleus. It's got a plus in it to remind us that the atomic nucleus is made out of protons and neutrons. And then there's this cloud of electrons that sort of flies around the atomic nucleus. Now, those electrons are organized. They're organized into orbitals, and then the orbitals come together into shells. The very outermost shell is called the valence shell. Sometimes there is an unpaired electron in a valence shell. This atom is unstable like this because electrons need to be paired in order to be stable. So if two atoms that have unpaired electrons come together, they can share their unpaired electrons, and now each atom has a pair. And this sharing, this bringing together, connecting these atoms, that's a covalent bond. Does anybody have any questions about that? It's such an important concept for us in biology because a lot of what we talk about in biology is breaking bonds and building bonds. Cells spend time and energy and precious resources building molecules. And then they spend time and energy and precious resources breaking the bonds in molecules. So it's critical that we understand exactly what a bond is. A covalent bond is just a pair of electrons. And in a living cell, those electrons are a source of energy. So that covalent bond is storing electrons while they're in the bond, while they're being shared. The energy that they have is in the bond. It's being stored. But if a cell breaks that bond and releases those electrons, now that energy has been released into the cell. Remember, the cell can't create energy. It can't destroy energy. All it can do is change the form that the energy is in. Oh, there's something else I want to show you on this diagram here. There's another type of bond that we talk about relatively often in cell biology, not nearly as often as the covalent bond. But there's another kind of bond that we see called an ionic bond. Sometimes when two atoms come together and each one has an unpaired electron in their valence shell, sometimes it makes more sense, again, in terms of which elements we're talking about. Sometimes it's more energetically favorable for those two atoms, instead of sharing their valence electrons, to transfer that unpaired electron from one atom to the other. This atom here, whatever it happens to be, is going to literally give up its unpaired electron over to this atom. Now we've solved the problem. There's no more unpaired electron in this atom, and there's no more unpaired electron in this atom. But they are not sharing those electrons. We had an actual transfer there. That means that this atom lost an electron and this one gained. Electrons have charge associated with them, negative charge. So this atom got more negative charge, and we now call it a negative ion or an anion. This one lost negativity when it lost its electron. So it became less negative or more positive. This one became a positive ion or a cation. So sometimes atoms share electrons. That's a covalent bond. Sometimes they transfer them. That's an ionic bond. These two atoms are still bound together, but they're bound together differently. It's their charge that's holding them together. So covalent bonds, very important to us in cell biology, but there are non-covalent bonds that are important too. Ionic bonds are one of them. And then there's two other types of bonds we need to be familiar with. These are non-covalent. One is called a hydrogen bond, and one is called a van der Waals interaction. Covalent bonds, the sharing of electrons, they are very strong, hard to break. Within a cell, there are actually different types of covalent bonds because they're in different molecules. For example, the type of covalent bond that holds the backbone of a strand of DNA, it creates the backbone of a strand of DNA, holds it together. That type of covalent bond gets a name. It's called a phosphodiester bond. Phosphodiester bonds are found in the backbone of a strand of DNA. In protein, the covalent bond that holds the amino acids together gets a name. It's called a peptide bond. In carbohydrate, the covalent bond that holds simple sugars together is called a glycosidic linkage. And finally, in phospholipids, the kind of lipid that you find in membranes primarily, the kind of covalent bond that holds that phospholipid together is called an ester linkage. There are 4 macromolecules in cells. Macromolecule meaning big, big molecule. There are 4 of them: protein, carbohydrate, lipid, and nucleic acid. In other words, DNA and RNA. Protein, carbohydrate, lipid, and nucleic acid. Protein has peptide bonds. Carbohydrate has glycosidic linkages. Lipids have ester linkages. And nucleic acids have phosphodiester bonds. They're all covalent bonds. They're just present in different molecules, so they get different names. Ionic bonds also exist in cells. Hydrogen bonds and van der Waals interactions are different because those two are quite weak. Covalent bonds are strong. Ionic bonds are less strong than covalent, but they're still pretty strong. It's hard. It's hard to break an ionic bond for a cell. Hydrogen bonds and van der Waals interactions are different. Those are weak attractions. We're going to talk, when we talk about the macromolecules, about where we find hydrogen bonds and where we find van der Waals interactions, because they're found in very different types of molecules. But those are the weak attractions that hold things together in cells. There are strong covalent bonds, and then there are weak bonds, weak attractions that hold things together. So covalent bonds and non-covalent bonds. And there are three kinds of non-covalent bonds that we need to be familiar with: ionic, hydrogen, and van der Waals. An ionic bond we've already talked about. An electron gets transferred from one atom to another, creating two charged atoms that are bound together. Hydrogen bonds, again, weak attractions, are going to form between what are called partially charged molecules. I'm going to talk about that in a minute. Hydrogen bonds form in molecules or between molecules that are polar and hydrophilic. We need to associate those two terms with hydrogen bonds. Hydrogen bonds got their name because usually one atom in a hydrogen bond is hydrogen. That's how it got its name. Van der Waals interactions are weak attractions between two molecules or parts of molecules. But van der Waals interactions form when molecules are non-polar and hydrophobic. So polar and hydrophilic for hydrogen, non-polar hydrophobic for van der Waals. If you can remember those, yes, if you can remember those, you'll be a long way towards remembering how they're going to behave.
With the hydrogen bonds,
Yep.
isn't that like have to do with like the dipole and which one's more polar, and like between the oxygen, hydrogen, oxygen wins the fight, which is why it's polar in the direction of the oxygen. So that's positive, the hydrogen ends negative.
Yeah, so everything you just said is correct. Everything you just said is correct. In biology, we don't tend to talk about the dipole moment very often, but instead we refer more to electronegativity. So if you look, for example, at water, water's a great example. What holds water together is covalent bonds. The oxygen and the two hydrogens are covalently bonded together. But because oxygen has a much higher electronegativity than hydrogen does, the electrons in those two covalent bonds, oxygen to hydrogen, the electrons are much closer to the oxygen than they are to the hydrogen because of electronegativity. Now, you're right, that's going to create a dipole moment. But what's important for us to understand more is that water molecule, which is neutral, its electrons are unevenly shared. They're not being shared equally between the oxygen and the two hydrogens. They're sitting in a location that is much closer to the oxygen than it is to those hydrogens. And that creates what we refer to as partial charges. Partial charges occur in a molecule when there is unequal electronegativity in that molecule. Okay. Now, I know it's a lot of information, and I'm going to go through that. I'm going to show you some images too to help you try to learn this terminology. It's the same science that you learned in chemistry. We just use slightly different language in biology than they tend to use in chemistry, because we're applying these concepts into a living cell as opposed to chemicals in a laboratory. Okay, so water, H2O. Water is what we call a polar molecule. Now, what makes it polar? Polar things have partial charges in them. Water is not charged. It doesn't have a plus or a minus on it or anything like that. But inside the molecule, there are regions of the molecule that are more negative and regions that are more positive. And that's because of the positioning of those electrons. Differences in electronegativity between two atoms in a covalent bond create unequal sharing of electron pairs that are sitting in that covalent bond. And that creates what we call a partial charge. So we're going to define polar. We're going to say that polar molecules have partial charges. Partial charges form when there is unequal sharing of electrons in a covalent bond. Let me show you a picture. Here's our water molecule. The big red ball represents oxygen and the two little white balls represent hydrogens. And this straight line that's drawn here, that represents a covalent bond. So there's a pair of electrons being shared in this bond and a pair of electrons being shared in this bond. Now remember, oxygen has a much higher electronegativity than hydrogen does. So the electron pair that's being shared is actually much closer to the oxygen than it is to the hydrogen. It's not sitting here in the middle of the bond. It's not sitting equally between these two atoms. It's actually sitting much closer to the oxygen. Electrons have a negative charge. They are negatively charged particles, right? So when they sit close to the oxygen, they bring their negativeness with them. This oxygen then becomes slightly more negative because those electrons are sitting close to it. We say that the oxygen is now partially negative, and we designate that with this symbol. This is actually a lowercase d or a delta symbol, and it's delta minus. That tells us that this oxygen atom is more negative right now than it normally would be, because there are two pairs of electrons sitting really close to it. If the oxygen is slightly more negative because it's closer to the electrons, that means these hydrogens are slightly more positive because they're farther away from the electrons. They are less negative than they would be normally. These are partial charges. They are not actual charges. They are within the molecule. They are regions within the molecule that are either slightly more negative or slightly more positive because of the positioning of those electrons. Polar molecules have partial charges in them. And partial charges form when the pair of electrons in a covalent bond are not being shared equally. And they're not being shared equally because those two atoms that are participating in the bond, they have a different electronegativity. So let's think about what that means on a practical basis. We said oxygen has the highest electronegativity, followed by nitrogen, followed by both carbon and hydrogen. So if a bond was between a carbon and an oxygen, there's a big difference in electronegativity. The electrons would be closer to the oxygen. So you're going to have partial charges there. That's polar. If I took two oxygens and I made a covalent bond between them, those two oxygens are both very electronegative, but they're equally electronegative. So they're pulling on the electron pair in equal and opposite directions. Two oxygens that are covalently bonded together, that pair of electrons is going to sit right in the middle of that bond, because this oxygen is pulling it this way and this oxygen is pulling it this way, and they're pulling equally strong. That bond has a pair of electrons that are being shared equally. So there's no partial charges there. And that means that's non-polar, no partial charges. So different electronegativities, partial charges. Similar or same electronegativities, no partial charges. That's what I want you to think of when you think of covalent bonds. You need to think about the two atoms that are in the bond. Do they have the same or different electronegativity? If it's the same electronegativity, that means they're going to share the electrons equally. The electrons are sitting right in the middle of the bond, and there are no partial charges created. That means we're talking about a non-polar covalent bond. If the two atoms are different from each other and they have different electronegativities, now you've got the electron pair sitting closer to one or the other. That creates partial charges. That's a polar covalent bond. That's the difference. So what about if I put a bond between a carbon and a hydrogen? I make a covalent bond between a carbon and a hydrogen. Two different elements, two different atoms. Where are the electrons going to be? Type into chat if the electrons are shared equal or unequal. Think about what we said about the electronegativity of carbon and hydrogen. Are the electrons going to be shared equal or unequal in that bond? Carbon and hydrogen. Good. Good. You got it. They're two different elements, carbon and hydrogen, but they have the same electronegativity for our purposes. It's actually slightly different, but in a cell, that difference isn't noticeable. It's negligible. Carbon and hydrogen have essentially the same electronegativity. So when a bond forms between them, they share the electrons equally. The electrons sit right in the middle, no partial charges. So that's a non-polar covalent bond. Good job. Remember, we talked about these weak attractions. We talked about hydrogen bonds and van der Waals interactions. Here's what I want you to remember. Hydrogen bonds form when partial charges are present. Jot that down in your notes. Hydrogen bonds form when partial charges are present. So that means hydrogen bonds form between polar molecules. Van der Waals interactions form when there are no partial charges. Van der Waals interactions form between non-polar molecules. That's what we need to understand in cell biology. When molecules come in the vicinity of each other, if they're two polar molecules, or even parts of molecules, two polar molecules that are coming into the vicinity of each other, they're going to hydrogen bond. If I have non-polar molecules or parts of molecules that are coming into the same region, they're going to van der Waals interact with each other. That's important because it's going to tell us how molecules will behave. Remember, we talked about DNA. We said DNA is a double-stranded molecule, at least in living cells. What holds those two strands together is hydrogen bonding. Remember, DNA is made out of a long string of subunits that are called nucleotides. And there are actually different nucleotides. Each one has a different part called a base on it. A, T, G, and C. There are 4 different bases. So there are 4 different nucleotides that make up DNA. Some of those bases can hydrogen bond together. They're all polar. The adenine base likes to hydrogen bond with the thymine base. They're both polar. And when they come near each other, they're going to hydrogen bond with each other. Guanine likes to hydrogen bond with cytosine. They're both polar. And when they come in the vicinity of each other, they will hydrogen bond together. That's what holds DNA together. The fact that those bases are attracted to each other because of their covalent bonding. So that's what we need to remember. Think hydrogen bonds, think polar. Van der Waals interactions, think non-polar. If you can remember that, you're in good shape. You're in good shape. That's what you're actually looking at on this slide. You're looking at 2 water molecules. Remember, these water molecules, in these covalent bonds, we have unequal sharing of electrons. That means that water has partial charges in it. Hydrogen bonds are going to form between molecules that are polar, that contain partial charges. The partially negative oxygen is going to hydrogen bond with the partially positive hydrogen. That's what holds water together. That's what gives water its surface tension, all of this hydrogen bonding. Now, down here, you're looking at the world's shortest piece of DNA. Here's one strand, and here's the other strand. And each strand has 2 bases jutting off of it. Here's a thymine base, and here's an adenine base. They're both polar. Can I look at thymine or look at adenine and know that it's polar? I actually can. Look at this thymine. Just look at the ring in thymine. Remember, when you see a ring on a molecule, that's made out of carbon. Those are carbon atoms that are bound together in that ring. So I have a lot of carbon in here. And look what else I have. Oxygen and nitrogen and a little bit of hydrogen. Would you predict that a molecule that has both carbon, hydrogen, and oxygen, nitrogen, would you predict that that's polar or non-polar? There could be oxygen to carbon bonds. There could be nitrogen to carbon. There could be oxygen to nitrogen. There could be oxygen to hydrogen. When you have a molecule that has all 4 of those critical elements in it, it's almost always polar because there's atoms in there that have very different electronegativities. In comparison, if I saw a molecule that had only carbon and hydrogen in it, I would predict that one to be non-polar because it's got only carbon and hydrogen and they have the same electronegativity. A molecule that contains only carbon and hydrogen can only make a certain number of bonds, right? Either carbon to carbon or carbon to hydrogen. And in both of those, we're going to have equal sharing of electrons, non-polar. So you can actually look at a molecule, even a really big molecule, and you can make predictions. You can say, you know, that thing looks very polar, or that thing looks very non-polar. Polar things are going to hydrogen bond with other polar things. Non-polar things are going to create van der Waals interactions with other non-polar things. We can make predictions about how molecules will behave in cells based on that much chemistry. Polar molecules contain partial charges. They like to associate with other polar molecules. They like to form hydrogen bonds with other polar molecules. We've said that. Now, remember this. The most universal solvent on this planet is water. Living things are made out of water, primarily water. Human beings have anywhere from 60 to about 75% water. All living cells contain water. So water is the solvent inside of a cell. Water is polar. Water is polar. So what does that mean? Well, it means that any other polar molecule is going to be attracted to water because water's polar. Polar things like to be with other polar things. Any molecule that is non-polar is going to be repelled from water because non-polar things don't want to be with polar things. They'd rather be with other non-polar things. So in a living cell, non-polar molecules are kept apart from the polar, watery environment. They're separate. They're kept separate. They're repelled and kept separate. And that's actually really important because it allows for compartments to be made inside of a living cell. If everything in the cell was polar, there wouldn't be a cell because it would all dissolve into the water. You have to have non-polar things in there that aren't going to dissolve into the water. So there is terminology around this. These words hydrophilic and hydrophobic are important to us. Polar things like to be with other polar things. Polar things like to be with water. So we describe them as hydrophilic. That word just means water loving. Polar molecules are hydrophilic because they like to be with water, because water's polar. Non-polar molecules do not want to be associated with water. Non-polar molecules are therefore hydrophobic. They are water-fearing. Non-polar molecules, they want to be with other non-polar molecules. They are repelled by water. So they are called hydrophobic. We talked about polar, non-polar. I put a couple of examples in here just to drive the point home. If you've ever made salad dressing, that's my favorite example. If you've ever made salad dressing and you've combined oil and vinegar, you know that polar things and non-polar things don't mix. Vinegar is polar, oil is non-polar. They're not going to mix with each other, are they? They're going to stay separate. They're not going to be able to bond with each other or interact with each other because they have two very different chemistries. And as I said a minute ago, that's actually good for life because in order for a cell to exist, it has to have some components that are separate from other parts of it. In order for for living things to exist, for cells to exist, they have to be able to be separate from their environment. A cell has a membrane around it. And that membrane is what defines the boundaries of the cell. If you look at the structure of a membrane, you're going to see that part of it is polar and part of it is nonpolar. That's what you're looking at in this image. This picture represents just a little piece of a cell membrane. Cell membranes, remember, have two layers. A monolayer and another monolayer come together to make a bilayer. Cell membranes are made out of phospholipids and proteins. If we just think about the phospholipids for a minute, we draw phospholipids as having a head, that's the circle, and two tails. That's what's hanging down from each circle. The head region is polar. The tails are nonpolar. So when all of these phospholipids come together to make a membrane, the heads are going to be oriented to wherever there's water. Because the heads are polar, they're hydrophilic. They like being around water. So they will face both the outside of the cell, which is largely made out of water, and the inside of a cell, which is largely made out of water. The tails of the phospholipids will be tucked in the middle. They are nonpolar, so they are hydrophobic. They need to stay away from the water. They can't mix with water. Those phospholipids, they're not bound together in a membrane. They're just associated with each other. They're attracted to each other. They are attracted to each other because of their chemistry. Those polar heads are going to hydrogen bond with the water. Those non-polar tails, they're going to form van der Waals interactions with each other. That's what holds membranes together. Their chemistry. The polar stuff likes to be with the water. They'll form hydrogen bonds. The non-polar stuff likes to be with other non-polar stuff. They'll form van der Waals interactions. Plasma membranes, phospholipids that make plasma membranes. We talk about them, we give them a special term. We refer to them as being amphipathic. Amphipathic. What that word means is any kind of a molecule that has two chemistries in it. It has both polar and non-polar regions. Protein is also amphipathic. Phospholipids have polar heads and non-polar tails. So they are amphipathic. They have both chemistries within them. Remember, the head of a phospholipid, we call it a head, but it's made out of a glycerol molecule and a phosphate group. The tail of a phospholipid is made out of a hydrocarbon chain and a carboxyl group. Remember, these groups, phosphate group, carboxyl group, these functional groups that you find in molecules, that's where the chemistry happens. That's where the reactivity is. That's what allows things to come together and react with each other. This last slide I put in here, I'm not going to talk too much about this today because we haven't talked about the macromolecules yet. So I'll bring it back up in a couple weeks when we talk about macromolecules. Okay. Any questions? I'm sure you have lots of questions. We're going to talk more about chemistry. We're going to keep bringing this chemistry up as we talk through these cell biology principles, because the chemistry keeps coming up. The chemistry keeps applying. If you can understand this much, that what we've talked about today, if you can understand this much of chemistry, of biological chemistry, you're in really good shape. Because this is the kind of chemistry that matters in biology. It's what helps us understand why things happen the way they do in cells. I mentioned a few minutes ago the idea that things bind to receptors on membranes. So remember, we said when a viral particle comes in and just randomly happens to collide with a receptor, it's because there is chemistry there. That viral particle, remember, it's surrounded by protein. And that receptor is made out of protein. And the parts of the proteins are going to be attracted to each other. Polar parts of protein are attracted to polar parts of protein, and they're going to come and they're going to bind together. That's why this chemistry matters, because it helps us make predictions about what's going to happen, I do see this one other question that Cecilia had said. When we were looking at that DNA molecule, the other base, remember I said, can we look at a molecule and predict, is it polar or non-polar? You're right. The other base had primarily nitrogen. So nitrogen, not oxygen in the other base. So the other base had carbons around the ring and nitrogen, not a lot of oxygen. Remember, nitrogen has a higher electronegativity than carbon and hydrogen does. So even if there's no oxygen in a molecule, but there's nitrogen in it, as well as carbon and hydrogen, you're still going to get partial charges because the nitrogen will pull the electrons towards it. Nitrogen has a higher electronegativity than carbon and hydrogen. Unequal sharing.
I thought you were talking about which one was more polar, like which
Oh
base.
yeah. You know, we don't do that. We don't talk about measuring polarity so much in this class. You can measure it. Some things are more or less polar than each other. Some molecules are more polar than other molecules. But that doesn't really matter so much in a cell, because what really matters is it polar? Is it polar? Because that's going to help us predict how it will behave in most situations. You're absolutely right, though, that when you take two polar molecules and you compare them, one of them is going to be more polar than the other one. But in a living cell, as long as they're both polar, they're going to be attracted to each other, and they're going to be attracted to water. That's a great point. All right, everybody, that's all I have for you today. Remember, you're going to want to watch that lecture video about the organelles. And if you have any questions about anything that you see in that lecture, message me through Canvas. Okay, send me a message and we'll talk about your questions. Otherwise, when you finish watching the organelles video, you're going to be ready to do your outlines. You're going to make 2 outlines, one for each lecture, however you want to make them, however they're useful to you for studying. And then you're going to submit them to me. Okay, so you can get some credit for them. I got your message, Mackenzie. Thank you, Mackenzie. I appreciate that. If anything else comes up between now and next week, you can also message me. No problem. Contact me if you need help with anything. And other than that, I will see you on Tuesday in the classroom. Hooray! Yay. All right, everybody, I'm going to let you go. Enjoy your evening. Have a good weekend. And I'll see you next week.
Thank you. Have a nice day.
You too.
Have a nice weekend.
You too.
Thank you.
Thank you. They have already addressed this, but one thing that I did notice is that you can't submit a PDF file for the, do you want it specifically in a docs, a Google Doc? not a Google Doc.
Actually in a Word document. You know what
Yeah.
I'll do though? I'm going to try to see if I can set Canvas to take the PDF.
Yeah,
but
because
if-
just that's the easiest and
Yeah.
only way I know of to get a picture on
Yeah.
there. So,
Let me try. And if I can't do it, I'll send an announcement out. Okay.
okay.
But we'll see if I can do it.
Thank
right.
you.
All right,
Bye.
everybody. We'll see you later.